and welcome to the second of two Storer lectures from our special guest today, Dr. Erich Jarvis. These lectures were made possible due to a gift from Drs. Ruth and Tracy Storer. Dr. Jarvis graduated from the High School for the Performing Arts in New York with a major in dance, so he's multi-talented, followed by a bachelor's degree in biology and mathematics from Hunter College in New York, and a Ph.D. in neurobiology and animal behavior from Rockefeller University. He began his faculty career at Duke University and is now back at Rockefeller as head of the laboratory for the neurogenetics of language. He's won some of the most prestigious awards in our field, including the NIH Director's Pioneer Award and the NSF Alan T Waterman Award. Dr. Jarvis is an expert on the neurobiology of songbird vocal learning in the context of the evolution of language. So last night, we heard about Dr. Jarvis's leadership in the Vertebrate Genomes Project, which has as its goal the sequencing of at least one high-quality genome from all 71,000 vertebrate species. And this afternoon, we're going to hear about his work on the evolution and mechanism of brain pathways for vocal learning and spoken language. So please join me in welcoming Dr. Erich Jarvis.
Thank you very much. Well, it's good to be back here again. I hope I can coordinate this well. It was hard to get used to a live audience I haven't talked to in so long, plus an online audience. So can they hear me on Zoom? Is that working well as well? Great, fantastic. So, here, today I'm going to talk about my original passion in science, which is about the brain. And when I got involved in sciences, I wanted to understand one of two things. I was trying to decide between understanding the origins of the universe, in which my mathematical background would take me, or how the brain works, in which my biology background would take me. And I chose the brain because it's connected to dance. And that's the main reason. But I think it's a journey that has been exciting for me, and I hope will convince you it's an exciting one to go for those who like to join this effort. So, but I chose to study a trait that I considered that was complicated, but not too complex, such that if we learn from a complex trait, the simpler traits would fall into place in understanding the mechanisms and the evolution. And that is a trait of spoken language. And it's a very rare trait. Only a few groups have it, or the ability to imitate sounds shown up here, and a few don't. And so I won't be able to get to everything today. So what I don't cover, you can read up on this article here that we published in a special issue in Science magazine a few years ago on the neurobiology of language, we and others. And so I have broke down this into 4 different topics. 3 of them is at the different biological levels of analysis, the behavioral, the anatomical, and the genetic. And then we'll end off with testing some hypotheses with some genes in mice. So at the behavioral level, when I say spoken language, what am I talking about? I'm talking about really one component that's more specialized. So what you see in the circle here is that what we humans experience every day as language, and particularly spoken language, consists of multiple component traits that we don't appreciate, but you recognize that they're components when you compare us with non-human animals. Some of these traits, like auditory learning or vocal usage learning, the ability to learn how to produce a sound, even innate sound in different contexts, is kind of ubiquitous amongst the animal kingdom. Whereas others, like shown up in here in red, vocal production learning is rare. And when we define it under these conditions, I define spoken language as a specialized form of learned vocal communication with some components found in most vertebrates, and this highly specialized component found in only a few species. And who are those few species? There are 5 groups of mammals. Us humans, of course, dolphins and whales, collectively called cetaceans, elephants and seals, or pinnipeds and broadly, and 3 groups of birds, songbirds, hummingbirds, and parrots. And all of these lineage of species have the ability to produce imitated sounds that they weren't born to be able to produce, and pass on those repertoires of sounds culturally from one generation to another. And all of them have close relatives that don't have this ability. So it's argued, like chimpanzees are to humans or falcons are to parrots. So it's thought that we 5 mammalian groups and these 3 bird groups evolved this trait independent of a common ancestor. What about another one, auditory learning? That's necessary for learning how to imitate speech, but not sufficient. This, I mentioned, is found in many species, like in your common pet animals. Like you can teach a dog to understand the human words sit, or siente se, osuwari. Does anybody know what language osuwari is? To sit? Japanese, okay? I bet you there are plenty of dogs know what it means, and you didn't, right? Or roll over, get the ball, and so forth. These aren't words that a dog learned to produce, and a dog can't say, okay, you got it, I will sit. No, instead a dog goes woof, right? And it can modify its woof to small degrees, but it's mostly in innate vocalizations. And so, but not all vocal learners are equal. Some are more advanced than others. The most advanced non-human vocal learner are parrots. And here's an example of a budgerigar, which is a small parrot called a parakeet, who was raised with humans for 4 years. And this is what Disco could do.
Never shake a baby bird. That would surely be absurd.
Never shake a baby bird. That would surely be absurd. Are you not a crook? My name is Disco. Cat, meow, meow. Give me a kiss. Hey, kid, has cheeseburger. Are you a little stork or a stormtrooper? Disco, I used to be a spy. Not a parakeet. Sure, I'm a doctor, not a parakeet. I did. I'm a doctor.
So that's amazing. I hope the people online heard that. If not, we'll play it towards the end. I'll turn up the volume. Here, Disco is not only knew or learned how to produce up to 400 words in his lifetime, but is actually recombining them into new meanings in different contexts. Many times, Disco is actually combining them and makes no sense to the listening humans, even if Disco thinks he's making sense. But other times, like he sees that dog and goes meow, kind of gets the category correct, but the actual species incorrect. But he thinks he's a doctor, as you heard. And so, so that's quite remarkable. And then every once in a while, a study comes out with one of our closest relatives, like Coco, a gorilla here, a great ape, where some scientists say that Coco is learning how to produce, getting something close to human speech. But this is the best Coco can do.
How about when you're coughing? That was good. You did a sneeze and then a cough. Excellent.
Yeah, so that's excellent, but it's not what Disco could do, right? Coco can't say, I'm not a gorilla, I'm a cook. I mean, not a crook, I'm a gorilla, or whatever, or a doctor. What's going on here is that Coco has very good auditory learning. Coco can understand several thousand human words auditorily, perceive them. Not as good as the 20,000 words a regular college-educated student has in his or her vocabulary, but 2,000 understanding is very good through auditory learning. Coco can't produce at least one of those words. This is the best Coco can do, is voluntarily control the air going up through the vocal tract, but not modulating the vocal tract to produce learned speech sounds. And so, so this has prompted myself and other scientists to say, how come our closest relatives, like Coco and others 6 million years removed from us, from a common ancestor, can't say something as simple as apple, whereas our distant relatives, these parrots, 300 million years removed from us, can go much further and say golden delicious and Macintosh and all different kinds of varieties of apples. What is going on here? And a number of people looked at, asked that question, and that's what I'm going to talk about in the second part of my talk, is what is going on here? What makes this difference that I'm going to call the motor theory of vocal learning origin? And, but my theory, or others, is not the only one that's around. There are multiple of them. And here is shown 6 popular ones of what makes this difference. In each of these diagrams, green is the forebrain, or the cortical areas, or in basal ganglia. Red is the brainstem motor neurons that control the vocal organs. And blue, the stars here, are the actual vocal organs. And one hypothesis is that the forebrain regions in the vocal learning species are bigger, or you have more neurons, and that then allows for more complex circuits for hearing or speech production and so forth to evolve. Another hypothesis is that it's not in the brain, it's actually in the musculature itself. One of them being that the musculature is more complex, more muscles in vocal learning species, or descendant larynx in humans that allows us to produce a greater variety of sounds that our non-human primates can't produce. Another hypothesis is that it's really not in the muscles here or brain size, it's the presence or absence of a particular neural network regardless of brain size. The haves and the have-nots, where they would have, vocal learners would have this forebrain circuit that controls the motor neurons directly. Another one is that non-learning and vocal learning species have a forebrain circuit up here, but the difference is that in the non-learners, it makes an indirect connection to the motor neurons here, whereas in the learners, it makes a direct one or an enhanced direct connection that allows the cortex to have greater control over the neurons that control the voice. Another one here, it's not the connectivity to periphery, but it's in the forebrain itself, where one hypothesis is that the non-learning species are missing a connection from auditory circuits like secondary auditory region to Broca's area, and that it's present there in humans and the other vocal learners, or that it's weak here in the red arrows and strong in the vocal learning species. And finally here, the sixth one coming from the linguistic community, where they argue that there's a language module here, abbreviated LM, that tells the auditory and Broca's and speech areas what to do, and that this high-level language module has all the complex computations of what language is supposed to be about, controlling the hands, the voice, and so forth. And the work that we've done and the work that I've scanned throughout the literature, I think supports a combination of three of these hypotheses, where yes, human brains are bigger to our body size relative to many other species. The vocal learning birds actually happen to have twice the number of neuron density in their cortical areas compared to the non-learning species. And there is seen to be the presence or absence, or at a minimum, an enhanced circuit in the vocal learning species that makes a direct projection to the motor neurons from the cortical areas that you do not find in the vocal learners. This other stuff about language module, direct projections between auditory and Broca's area, I'm not convinced. Actually, I think the evidence is against it, including the descent of the larynx, which I don't have time to get into all the details of that today, but I just want to communicate that message. And so let's dig further into these hypotheses here, particularly the presence of a forebrain circuit. How did that come about, and how it functions was at least my contribution to that is what we discovered is that when songbirds sing, like a learned canary song like this, that act of singing is associated with a rapid increase of what we call immediate early genes in the white signal here, the mRNA product, in certain brain regions that control this vocal learning behavior. What you see above these birds here are brain sections. This is the front of the brain with the forebrain up here, and this is the back of the brain with the cerebellum, and the red stain here is just a general stain for neurons. And what we see is that when these birds sing, in the white signal here, the messenger RNA product, like of CFOS or EGR1 genes, comes on at very high levels, such that for every bout the bird sings within a half-hour period, you get a one-fold increase of the messenger RNA product of these genes. It's a very energetic, robust response. And this occurs in a motor-driven fashion. And that is when these birds are hearing other birds sing, you don't see this activation in these brain regions like area X and HVC. You don't see it when you remove somatosensory feedback, or you play song or you look at a bird and so forth. You see it only when you actually try to move your muscles. Even a mute bird where it doesn't have the connection, sever the connection from the motor neurons that control the larynx is there. The bird tries to mouth the vocalizations, you still get this activation here. So we call this a motor-driven gene expression response. And what we did is we used this approach to map vocal communication circuits across species like an MRI tool. And we found that whenever birds heard song, you show these areas here that are lighting up in blue, or blue labeled areas that were lighting up with hearing song, you can find them in all bird species, whether they're vocal learners, like here, songbirds, hummingbirds, and parrots, or non-learners, like a chicken or a pigeon. Consistent with the idea that the auditory pathway in the forebrain was inherited in vocal learners from their common non-vocal learning ancestors. And this is why we think all bird species have auditory learning, and mammals as well. However, only the vocal learning species had in red and yellow here these brain regions that lit up with this gene expression activity, and when you put electrodes, they fired and so forth, when they're producing their learned songs. And there are exactly 7 of them in each of the vocal learning bird groups. And in red, we knew from the work in songbirds and parrots that this is the circuit necessary for learning how to imitate the sounds. It's what's called the cortical basal gangliothalamic loop. And in yellow is the brain regions that project out of the forebrain, synapse onto these gray structures here, which are the innate brainstem structures that control innate vocalizations found in all species. And this motor pathway then controls the production of those sounds and has taken over this innate brainstem circuit. So looking at this tree, we had concluded this tree next to here, next to these bird brains, is that either there were 3 independent gains indicated by these red dots in the last 65 million years, and each time the vocal learning circuit evolved similarly. Or there was a common ancestor back here, followed by 4 independent losses, and only these vocal learning species are the lasting ones that had maintained the circuit. In other words, it would be like humans and chimpanzees, or and gorillas having a common ancestor with spoken language, and humans maintaining it, and chimps and gorillas losing it. So that's what that model would be like. Or everybody has it to various degrees, and it's independently amplified in these vocal learners. And the techniques we use, we haven't been able to detect it in these species here because it's so small. Or maybe this phylogenetic tree is wrong. The tree that I have been using for 15 years since I was a graduate student to try to understand the evolution of vocal learning. But what am I, a neuroscientist, to tell the phylogeneticists that they got the tree wrong? But when I showed people this result, they said, this got to be a common ancestor here. Another hypothesis, I think I mentioned this yesterday as well, that was proposed is that when we first came out with these studies, some religious groups took these pictures, put it in online religious texts to say this helps prove intelligent design. All right, and I didn't have a good hypothesis for that just yet, and we'll see that we don't. Well, anyway. I just want to entertain that possibility because it's been said. All right, so and as you heard, we got involved in genomics, and we ended up sequencing the genomes of all of these different species here and some more, representing all bird orders. And for the first time, did a phylogenetic tree based upon the entire genome, as opposed to different genes, which can actually result in different tree structures, and came up with this new tree here that showed actually lots of rearrangements according to the tree that I had been using for 15 years. But what about the vocal learners, shown here in the red dots, is that it actually brought the hummingbirds much further away from songbirds and parrots than we had in this older tree, supporting the idea of independent gains. If we had to have a common ancestor, it would have to gone back here at the time of the last mass extinction of dinosaurs 66 million years ago, followed by 10 independent losses throughout the avian family, or the Neo-Aves part of the tree, which we think is highly unlikely. And so, we convinced ourselves that this trait is independently evolved in the vocal learning species, despite the presence of 7 similar brain structures with a similar circuitry. Not identical. What about us humans? Well, if these birds came up with a similar solution over the last 65 million years, have we humans in the last million years did something different? And so we began to ask that question first through comparative analysis of the literature and just simple, principles here. Here is a zebrafinch brain, and it has no cortical folding and so forth. We doesn't think that matters. And here is a zebrafinch brain. Here you can see at the top to your right here to scale with a human brain. You can fit 3,000 of these zebrafinch brains into a human brain. So neither cortical folding nor size matters in this case. I argue it's the presence or absence of a particular neural network that matters. And to get at what that neural network is, doing a lot of comparative analysis of what we know about speech brain circuits and comparisons with non-human primates and with these vocal learning birds, I came up with the following hypothesis that's been developing over time. This is the latest that we've view of this. I argue that we humans have inherited a Wernicke's auditory area involved in speech perception or complex aspects of sounds from a common ancestor with all vertebrates, not just of primates, not just of mammals, but all vertebrates. I argue that chickens and pigeons and cocoa has a Wernicke's area, so does your pet dog, and this is why they understand sit, siéntese, osuwari, get the ball, how about coughing? And that we inherited this ability from our common ancestors. But only humans amongst the primates evolved this similar circuit we find in these vocal learning birds that consists of a dorsal and ventral laryngeal cortex region that has taken over these innate brainstem circuits in the human brain, found in non-human primates, for producing learned speech, and these more interior forebrain cortical areas like Broca's, the pre-laryngeal motor cortex, part of the basal ganglia, anterior supplementary motor area that's involved in more complex aspects of speech production, syntax, and vocal learning. And so how could that have come about? Because when we published that picture, it also went into religious texts, because now it's 300 million years removed from a common ancestor. And I didn't have the answer until we came up with this discovery, where we found that not only do you see this immediate early gene activation in these song nuclei here with the learned singing behavior, but when these birds are hopping around in the rotating wheel for the very first time and learning how to coordinate their bodies, we get this movement-driven gene expression, this motor-driven gene expression surrounding all the vocal learning nuclei or adjacent to them, all 7 of them, in all 3 vocal learning lineages. And we did the same experiments on the non-vocal learning species, and we found the movement-activated brain regions, but with no holes inside of them where we would expect to find song learning nuclei. So pigeons, quails, and ducks, and so forth had the surrounding regions, but not the song nuclei. And when we look at the connectivity of the song learning nuclei themselves and the surrounding brain areas, we find that there are parallels. In white arrows here, there's cortical basal ganglia thalamic loop for the surrounding motor circuit, the descending motor pathway as well. Differences is that it didn't make a direct projection to the motor neurons for controlling limb muscles. It made indirect projections. For those that know about that, that's what's more common amongst vertebrates. What's interesting is that the RA nucleus had collaterals, the surrounding motor areas, it's shown in this red arrow, had collaterals going to the striatum here in purple, whereas the vocal learning circuit did not. And this is more commonly found in motor circuits of mammals, where you had these collaterals going to the striatum from the descending layer 5 neurons. And so this indicates some type of ancestral relationship of the vocal learning circuit with motor pathways. And around that same time, getting back to my past lifetime, Steve Brown, a colleague of mine, was trying to study movement learning circuits in human brains for dance. And he would take well-trained dancers, get them to do learned choreographed sequence movements, and would find that some of the areas of highest activation, like here and here, were adjacent to laryngeal motor cortex and Broca's area, that is the speech production areas. And so putting all this together, I came up with the following hypothesis or theory called the motor theory of vocal learning origin, where I argue all species of vertebrates that vocalize have this innate brainstem circuit in the midbrain and the hindbrain that controls innate crowing, innate dogs barking, and so forth, or our cries, our screaming. All of these vertebrate species, I argue, also have a forebrain motor learning circuit that is replicated during embryonic development to connect to different motor neuron groups to control the arms, the legs, the wings, the chest, and so forth. And that in the vocal learning species, I argue through some type of gene mutation or gene duplication, this motor learning circuit is duplicated like a whole brain pathway duplication one more time. And that new duplicated circuit synapses onto the brainstem circuits for vocalizations and takes it over to get this emergent vocal learning circuit that is similar to the surrounding motor pathway. Think of it like gene evolution. You can duplicate a gene, and you have an old gene that keeps the normal function and a new gene that becomes neo-functionalized for a new function. And this is what I argue could be happening for whole brain circuits. So how we're going to test this hypothesis, or in general, the relationship between these brain regions, and that, I thought, is that if this is true, the genes that function inside of these vocal learning brain regions should be genes that function similarly in the surrounding brain regions or in motor learning circuits, except for differences in neural connectivity, differences in maybe how the vocal musculature needs to be controlled, or neuroplasticity. And so, so that was the major goal of the next stage of my long trajectory here and what I'm trying to do with my career. And this has also brought me back into genomics, which is looking for the genetic convergence. If the circuit and the behavior is convergent, are the underlying genes convergent in these vocal learning birds and with humans as well? And that motor theory of vocal learning origin argument could apply to humans as well. And to get at this, when we first started doing this research, microarrays, for those that heard about that at the time, were the popular thing. And what we did is we laser dissected out the song learning nuclei, like HVCRA, area X here, from each of the vocal learning species and their surrounding motor areas, popped them up to Eppendorf caps, isolated the RNA, and profiled them onto oligoarrays to profile gene expression levels in these brain regions relative to the surrounding motor areas. Allen Brain Institute was doing something similar for humans and non-human primates, so 3,700 samples from 6 people, several hundred samples from 6 monkeys, profiled them onto microarrays. And folks in my group at the time, who actually now gone on to their own labs, what they did is the bird wet lab work, as well as Andreas developed new algorithms to take this data, align up the genomes to get the correct orthologs across all these species, and then look for specialized expression patterns that are shared or not shared between species. And make a long story short, we found at this time, out of 7,000 orthologous genes between humans and birds that we can find expressed in the brain, of all the brain regions that the RA saw nucleus could match in a human brain, in the spinal cord, the basal ganglia, and so forth, or area X that is, it had an expression profile that matched an anterior part of the human striatum that was later discovered to be the most active region of the striatum involved in speech production or speech learning. The RA nucleus matched a part of the laryngeal motor cortex involved in production of learned speech. And the same brain regions make these direct projections shown by these red arrows for controlling production of the vocal of sounds to the vocal organ. And we did not find such areas in chickens or quails or macaques that had the same specialized expression as we found in humans or songbirds. In other words, humans and songbirds were more similar for these genes in their regulation than they were to primates, non-human primates. And when we look at the laryngeal motor cortex and the RI nucleus for those genes, it wasn't all 7,000. It was 55 genes in the RA cell nucleus. So shown here is a heat map where each row is one gene. Each column is a sample from, let's say, a songbird and a parrot here and a hummingbird. And what we can see for downregulation, blue, upregulation in red, that the pattern here in the vocal learning species is more similar to the pattern here in the human speech motor cortex than it is to motor regions of non-vocal learning species or other cortical areas not involved in speech. And I'm not showing the primate data here, but the primate data looks like the non-speech areas in terms of its gene expression patterns for these 55 genes. And so microarrays, we know, are subject to false positives. And so we try to validate some of these by in situ hybridization. Actually, before I get to that, I do want to say that what was interesting is that a lot of these downregulated genes were axon guidance molecules, like SLIT1 over here and plexin C1. And the upregulated ones were involved in like neuroprotection, like parvalbumin. And so some of these are genes that people have heard before, And that the down rate or the axon guidance molecule ones could make sense because of neural connectivity differences. Why upregulate these neuroprotection genes? I think the reason is that the larynx muscles and the syrinx equivalent in birds are the most rapidly firing muscles in the body. So you need fast-firing neurons to control these muscles, even excitatory fast-firing neurons, not just inhibitory ones. And when you stick electrodes into these vocal learning brain regions, their even spontaneous firing rate is higher than the surrounding motor areas. And so this is why I think we need these calcium buffering and glutamatergic and heat shock proteins to protect us from too much toxicity in our brain from talking. And so, we validated some of these by in situ hybridization. So again, the white signal is a messenger RNA product, showed downregulation of SLIT1 in all the vocal learning bird groups, but not the non-learners. And here in the human brain and parrots, we also found 2 brain regions each throughout the circuit, particularly in parrots, but also in humans, that some people for humans were arguing which was the proper laryngomotor cortex, this one here or this one here. We found actually both. That humans actually have 2 laryngomotor cortex regions, so do parrots, that actually correspond from work from Eddie Chang's lab to show that in patients undergoing surgery for tumors or epileptic regions, they put electrodes on the surface of the brain or into the brain to try to identify the speech areas so they don't actually dissect them out accidentally. And what Eddie's group has found is that these regions where we found convergent gene expression with birds are the 2 hotspots of increased neural firing during speech production in the motor cortex. And so we had a functional parallel here. And so, so we got all excited with this. Now we had 55 genes for RA and 75 genes for the basal ganglia. And now we're going to take some of our favorite genes like SLIT1. And finally, we found the convergent gene. We're going to study it and study its function in vocal learning circuits to discover that the gene is highly fragmented. Here is a Sanger base assembly. Each one of these dark and light brown lines here means there are different contigs across this 35 exon gene. Some of them are in what we later discovered are the actual protein coding sequences. And this is not the only gene. We start to find that in these short read-based genome assemblies, that many of them were missing parts of genes or entire genes. Some of them were incorrectly assembled. We found interesting repetitive regions in front of some of these genes that are specialized in vocal learning circuits. They were not assembled correctly once we did single molecule cloning to figure that out. And this was causing my students and postdocs in my lab and other labs to take many months, sometimes more than a year, to reclone these genes individually and through PCR reactions and sequence them the old-fashioned Sanger method to get the correct gene structure. And that just really made me upset. And so, so we were unknowingly working with artificial gene structure and sequence, and us and many other people were making false conclusions about the biology, like of gene duplications and so forth, that weren't true. And so while all this neuroscience research is going on, and I realized we need higher quality genomes, and as you heard yesterday, I was asked to take over leadership for the Genome 10K Consortium because of our success with the bird genomes. And what I ended up helping the group focus on is not just to generate many genomes for our particular questions, but to help work with the genome sequencing companies and other experts in genome assembly to achieve a mission to generate high quality, error-free, near gapless, chromosomal level, well-annotated, and haplotype phase, which ended up being very important to get correct genome structure for not 10,000 species, but for all 70,000 vertebrate species, to utilize and address fundamental questions like I'm interested in, you're interested in, or conservation and disease and so forth. And so you heard about the outcome of that story yesterday, and I told you some more about it, but I'm going to tell you even more now. I can't tell you everything, but there is a special issue that we published on the initial publications or studies in Nature last year. I encourage you to go look at. And so how did it help our story here on understanding vocal learning circuits? Well, one thing is, if you're going to compare bird brains to human brains, you have to get the homologies correct. And and something I mentioned yesterday, and I won't go into further, we further, through RNA-seq profiling, with these more complete genomes and additional genes, have changed our understanding of the organization of the bird brain. The cortical areas consisting of 4 major cell populations, and here is the basal ganglia, the striatum in purple and the globus pallidus in blue. Keeping that in mind and looking at the human brain, all these different colors up here in green, orange, and yellow correspond to what we think are different layers of the mammalian cortex. And keeping that in mind, what Greg, a graduate student in my lab, found, he laser dissected out the song learning nuclei again and did now RNA-seq profiling as opposed to microarray profiling, and found doubling to tripling the number of genes that were specialized in, shared between humans and songbirds. So for this area X1, instead of 75 genes, we got 118 for part of the anterior putamen next to the anterior caudate for 86 genes. And for the RA, we confirmed that convergence. Actually, didn't turn out to be there were 2 laryngomotor cortex regions I mentioned before. One of them we find 42 genes that it's convergent with, and now HVC, which we couldn't find any result from before. Now that we had additional genes, more complete genomes, and surrounding brain regions, we found convergence also with the dorsal laryngomotor cortex for 116 genes. And when we add them all up together, we get several hundred genes for these cortical regions. And so, by the way, for the L-man song nucleus in birds and Broca's area in humans, they were both unique to each species. I think they function similarly, but we couldn't find overrepresentation of convergence. And so Broca's area seems to be more unique in terms of its gene expression profile, in terms of, for vocal learning species. The 2 brain regions of songbirds matching 1 brain region of human made us think, could these be different cell types? And so what you see in this heat map here is that of these several hundred genes that are matching between HVC and RA with the laryngomotor cortex, a small minority of them are up or down regulated in the same ways in HVC and RA. The rest of them are different, indicating perhaps different cell types. We did bulk RNA-seq for HVC and RA, so we didn't know whether or not they were different cell types. But fortunately, the Allen Brain Institute had taken some human motor cortex data and profiled different cell types from layers 2, 3, layer 5, layer 6, and so forth. And we took our specialized pattern of genes from HVC and RA and see if any of these different cell types in the human motor cortex match HVC or RA, and the answer was yes. HVC matched layers 2 and 3 broadly of its profile of the human motor cortex and layer 6. The RA song nucleus matched layer 5 neurons, which makes sense because RA and the human laryngomotor cortex had these layer 5 neurons that project out of the forebrain that control the production of sounds. And then we asked, within the songbird brain itself, Michael Brainard's group had actually profiled the individual cell types within HVC, and we compared that data with ours and found that the RA neurons that project out, the HVC neurons that project to RA is a specific layer 3 type neuron that is convergent with humans. All right? And for the RA song nucleus, it was the neurons that project out of the forebrain synapse onto the motor neurons for controlling the voice that was convergent with layer 5 neurons of human laryngomotor cortex. In other words, it's the projection neurons that project from one brain region to another brain region, not the local interneurons, but those projection neurons that are showing convergence between humans and songbirds for speech circuits and song circuits. And so, so that is, that's a great benefit of what we got out of these higher quality genomes. And then what about, we now begin to ask the question about regulation. What is causing the specialized regulation of these genes in these brain regions? And what we did is we laser dissected out the brain regions again and did ATAC-seq profiling. And for the genes that were regulated by singing or hopping equally in the surrounding brain regions, we got new promoter regions that we couldn't have sequenced before. And I mentioned this yesterday, but show it again today. we have ATAC-seq profiles in front of these genes in all brain regions and in liver. It doesn't matter. Wherever neural activity is regulating this particular set of genes, we see these open chromatin peaks. However, for genes like the androgen receptor, which was upregulated in the song learning nuclei, not in activity-dependent manner, but just endogenously higher, we found those kinds of genes had peaks in their open chromatin areas in front of the regulatory regions of the genes, or enhancers or introns, that were higher than the surrounding brain areas, indicating that this could be the regulatory region that's upregulating androgen receptor. And so that's just an example of 2 genes. But when we take all several hundred genes that are specialized in these brain circuits for song, we can find for HVC, for RA, for area X, that there's an enrichment of ATAC-seq peaks in their enhancer or promoter regions that you don't see in this other genes that are not specialized in the vocal learning circuits. So there's a nice correlation here of specialized regulation, regulatory peaks that we're seeing in their promoter regions or upstream enhancer regions. And when we look at these enhancer regions and look at the actual DNA sequence and ask, do we find transcription factor binding sites? And the answer is yes. We have a high prevalence, like for genes that are regulated by neural activity, or sorry, let me go the other way around. For genes that are upregulated in the song learning circuits, we find that they have 50% of them, 50% have the androgen receptor binding site. For genes that are turned off in the song learning nuclei, roughly 30 to 50% have the neural D6 transcription factor binding site. And for genes that are equally regulated inside or outside the song learning nucleus, sorry, inside or outside, have equal representation of the EGR1 transcription factor binding site for the activity regulated genes. And now then, when we look at the functional categorization of these genes, their biological functions, we find that using a human phenotype database, that many of them are involved in axon guidance, cell migration, cell motility, whether they are upregulated genes or downregulated genes. However, when you look at phenotypes, behavioral phenotypes, what's interesting is that these upregulated genes in humans, when mutated, are associated with speech deficits like poor speech or autistic components of speech in autistic children and so forth. And so when we put all this together, yes, there was one more aspect of this. Now then we start to dig down into the actual individual sequences, and what we find is when we, again, profile the ATAC-seq peaks of these genes, like for NeuroD6, the transcription factor that could downregulate some genes, we find upstream of its promoter as well as further up here in what we think is an enhancer region, accelerated evolution of rapid nucleotide changes in this part of the genome in songbirds, parrots, and hummingbirds, only the vocal learning species. And here is that particular sequence here. Here is this very conserved sequence found in all the bird species, but the vocal learners showing these red changes. There's an identical sequence in the vocal learners, and there's a unique one specific to each one. So both things, absolute convergence and parallel evolution here, that takes this sequence, we don't know what its function is, but just these 2 nucleotide changes in each of the vocal learners converts the sequence into an androgen receptor binding site, coincidentally, right? What is the NeuroD6 transcription factor doing with an androgen receptor binding site in front of it, only in the vocal learning species, in a brain region where the androgen receptor is very high? And by the way, the androgen receptor we found is also very high in human laryngeal motor cortex of both men and women. So that's an interesting paradox I didn't expect to find out. And I'd be curious to hear people's thoughts about why that might be the case. So summarizing this part of the talk, putting this model together where these tick marks here mean that only the vocal learning species acquired some mutations in front of the NeuroD6 gene, and that mutation then converts this conserved sequence into a binding site for a repressor, like the androgen receptor, that then turns off NeuroD6, and by turning off NeuroD6, you won't regulate its target genes that are also off in the vocal learning circuits, like SLIT1. And then how do you prevent turning off everything in the brain for neuro-D6, well, then you acetylate that new enhancer region so in the outside brain region, so it's only regulated in the vocal learning circuits. And then you can have other interacting partners, some can understand in this audience, Megan, for example, where this gene slit and its receptor, robo, interact with SRGAP2, which we know has an extra duplicated copy in humans, and maybe the whole thing is interacting in speech circuits to modulate speech circuits in a more advanced way in humans. So that's the model I'm thinking of. When we try to put all of the converging evidence together, we find that there are 26 genes for HVC and RA that have convergent gene expression with human laryngeal motor cortex, that have atax-seq peaks in their regulatory regions with these transcription factor binding sites of one of those 4 that I told you about, and then that when have mutations in them are associated with speech deficits. And just can't be total coincidence, like dysarthria, ataxia, and so forth. And one of those, some of them are example genes are the androgen receptor, ZEB2, another transcription factor, NFX, another transcription factor, plexin A1, an axon guidance ligand, SLIT1, an axon receptor, and the SLIT1 is the ligand, GRIN2B, even a glutamate receptor. And so involved in neuroplasticity. And an example of a mutation in one of these genes in humans is shown in this child, Bella, who's a 7-year-old child with a mutation in the ZEB2 transcription factor. And here's what her behavior is like. She's trying to say bubble.
Bubble.
So she can only say one word at a time. She has okay coordination for other behaviors.
What are you gonna
as
make?
you can see with
No,
putting this bubble blower into the bottle. And so Bella's auditory learning is pretty decent. Bella has a comprehension of thousands of words, all right? There are other deficits that she has, but the vocal deficit, like with a FOXP2 mutation, has a stronger effect. And so this is one of the genes that we'll be studying in the future. Another gene, like I said, GRIN2B was a surprise to us. And at the time, we knew that this gene in mammals and birds, and particularly in vocal learning circuits, is high during juvenile development and is turned off or downregulated to lower levels throughout the brain. But in the vocal learning circuits, like in L-MAN, HVC, and RA, it's even turned down even further, which we think is crystallizing the song to allow the bird to not learn new songs and forget. And so before we had a more complete zebrafinch genome, we used the human NR2B gene, we cloned it into a viral vector hooked up to the GFP protein, injected it into L-MAN here, and then asked the birds what happens. And what we found is that when you overexpress the human NR2B in L-MAN, the bird song becomes more variable. The more overexpressed you have, the more variable sequences you have or acoustic structure of your vocalizations compared to a scrambled knockdown. A knockdown with not knockdown, with just GFP only. Unfortunately, we had a side effect. So they became more juvenile-like, but they also stuttered, particularly when they were singing to females. And we don't know why that's the case. Maybe just manipulating one gene is not good enough to reopen the critical period plasticity here. Going forward, in terms of manipulations, what we'd like to do, if all of this is true, is those molecules that were, say, downregulated in the axon guidance molecules that were downregulated in these speech and song areas, what if we prevented that? Would we prevent the direct projection from forming and block vocal learning abilities? What about a non-learning species? If we actually downregulated here, took over a pre-existing motor pathway and got it to make a direct connection to the motor neurons for vocalizations in a chicken, and you get a chicken to start imitating. We would love to do that. The technology for that is not as advanced for birds as it is in mice. And so this is what we started to do now with these more complete sequences, is now we can go start testing some of these hypotheses in mice. And that's what I'll end off in this last part of my talk here. And we knew at some point we would be working with mice because of their ease of genetic manipulation. So we actually started these studies a little while ago just to even characterize their behavior and vocal communication circuits. And when we started this, Tim Hawley's group at the time came along with this study to find that mice produce these ultrasonic, particularly the males, ultrasonic songs. Here, like this male mice is producing to this female here, and here's the sonogram below, that sounds like a songbird song. And that really fooled us. We thought, oh my goodness, did we miss a vocal learner in the laboratory? It turns out that many of us and many people, including us, studied these mouse behaviors and found that actually it's not learned, although it's not totally innate. There's a small level of plasticity from work from my former graduate student here. We also, like in the song-learning birds, after getting the mice to sing, look for activity-dependent gene expression in the brain, and to our surprise, we found the motor cortex region with increased activation of these immediate early genes when these mice sing, basically, which was thought to be a cortex region for vocal communication was thought to be only present in vocal learners and humans amongst non-human primates. Also a striatal region where we'd expect to find area X. And when we did tracing studies using transsynaptic tracers injected in laryngeal muscles, we found that same area had layer 5 neurons in this motor cortex region that was active during vocalizations. And we placed tracers into that motor cortical area. We found axons that went down to the motor neurons here in brown, so the axons are black, making actual direct contacts to the motor neurons for the vocal organ, for the larynx, which was surprising, again, thought to be only unique to vocal learners. However, there was a difference. In the vocal learning species, like these birds here, or in humans, all these squiggly lines here coming from degenerating axons in a person who had a lesion in the motor cortex, including speech areas, you find that there are hundreds, dozens to hundreds of axons synapsing onto the motor neurons for vocalization behavior in the vocal learning species, songbirds and humans. Compared to mice, it's only 2 to 3 axons per motor neuron. So it looks like a difference, not an absolute difference here, but a difference of degree. What about the genes themselves? Like SLIT1 that's downregulated in the vocal production nucleus of songbirds and humans, we don't see that downregulation in mice. And so we're thinking more along a continuum here, like Roger Lemon has proposed for the limbs musculature, that coming from these axons coming from the cortex, many of them make these indirect projections, and as you get more and more advanced behavioral, at least coordination movement with your hands, you get more and more direct projections from the cortical areas and more connections coming from the cortex to control the musculature here. And shown here in a diagram form of what that would look like for humans, here is the laryngeal motor cortex. If primates really have one, it's making only indirect projections, whereas in humans it's making both indirect and direct projections to what's called nucleus ambiguous that innervates the larynx. So when we published this, some of the speech experts, the neurobiology of speech expert says, I don't believe you until you can actually stimulate this brain region in the mouse and show me that the larynx will vibrate. And so Cesar Vargas in the lab did just that, and he stimulated throughout M1 motor cortex and M2 in the mouse brain and found 2 regions. One, this putative laryngeal motor cortex area where we had those layer 5 neurons, showed a 10 millisecond delay and activity in the laryngeal muscles as well as the jaw muscle. And vice versa, there is an oral facial motor cortex region already known in mice, known to connect to the jaw muscles. It showed a pretty robust response, even with a shorter delay, and a weak response for the laryngeal muscles. So there's some interaction going on there. And when we noticed there was a difference between the strength of the stimulation in the cortex given. If you give 1 pulse into this more posterior region, more medial kind of like, lateral, I'm sorry, you get activity of the laryngeal muscles, but not the jaw muscles. And if you go to the more interior region, you get the oral facial motor cortex stimulating the jaw muscles, but not the laryngeal muscles. You have to give 4 pulses to give activity in both. So there is a separation between jaw and larynx, but it's not totally separated. And when we actually did transsynaptic tracing from both the jaw muscles and the larynx muscles, we found that in the forebrain here, there were neurons in green and red here that came from the muscles independently, but there were a few in yellow, a small minority in yellow, shown quantitatively here in gray, indicating that there were single layer 5 neurons that send one axon collateral going to muscles to controlling the larynx and one controlling the jaw. And when we did the same thing, I don't have time to show you, putting tracers in the hand and the jaw, we found even less, we found a few rare ones, but the hand and the larynx muscle are more separated in the mouse motor cortex than the larynx and the jaw. So, so putting all this together, and what we think is that it's a continuum, that there are species that don't vocalize, like lizards, that don't have vocal learning because you don't vocalize, right? Others, like mouse and chicken, that do vocalize to a limited degree, have vocal plasticity to a limited degree, followed by, say, the great apes and so forth with more moderate vocal learning abilities, to the complex vocal learners, the classical ones we talk about, like songbirds, parrots, and bats, and the more advanced vocal learners, like parrots and humans, who have 2 parallel pathways for vocal learning. And so it's not a black or white world. There's lots of shades of gray in between. With this hypothesis, if it's true, it would mean that maybe we can take a pre-existing circuit that's in the mouse brain with just 1 or 2 axons per motor neuron and influence it to make many more connections to the brainstem and have cortical control of its vocalizations that are stronger. And the idea for this comes from, actually we proposed it, but then Yoshida's lab published an idea on this for the limb motor neurons, where they found that human plexin A1 was at higher levels in mouse motor cortex and turned down in human motor cortex broadly. And they hypothesized plexin when it binds to its ligand, it actually is a repulsive interaction. So if you downregulate the gene, you will actually allow a connection to form that normally doesn't form. And what they found by downregulating plexin in the mouse motor cortex, in these red little dots here, they were able to get more axons coming from the motor cortex to synapse onto the limb motor neurons, and these is shown here, and these mice, they ate spaghetti faster, all right? They had more manual dexterity with their hands. And so what is that like for speech circuits? What we think is, so in the non-vocal learning species, you have high levels of plexin or SLIT1, and when these axons from layer 5 go down to the brainstem and they find the motor neurons with high levels of their receptor or ligand, they avoid it. It gets repelled. However, in the vocal learning species, we hypothesize that the ligand SLIT1 is turned down or the plexin receptor is turned down. Whether you have a receptor here or not, you don't avoid it, and you can now form a direct projection. So what Lomax Boyd did here, postdoc in my lab recently, some unpublished stuff, is we got a Cree line that had a promoter expressing Cree in layer 5 neurons, crossed that mouse with a knockdown of plexin as well as SLIT1, and were able to knock down plexin expression specifically in layer 5 and leave the rest of the brain untouched, okay? And what we found in these mice is that here in wild type, here are the axons in red coming from the motor cortex. You can see where nucleus ambiguus is located, only 1 or 2 axons are growing across. However, when you knock down plexin in the layer 5 neurons, and I'll blow it up here, you see many more axons going through nucleus ambiguus compared to the wild type. And when we look at quantifying this, the heterozygotes have an increase of innervation, and the double knockouts, the homozygotes, even have a doubling there. And when we look at the behavior, we find that these mice, compared to wild type, have more rapid maturation of the pitch of their vocalizations. And now we're testing if they actually have any vocal plasticity level. And stay tuned, you have to invite me back for that study. All right, I'm going to finish off with one other gene manipulation. I can't go a talk without talking about FOXP2. And here what we found is that Many of you know about this gene. It's also a transcription factor. When there's a heterozygous mutation in humans, this kind of deficit happens.
Your name?
Laura.
She's trying to say Laura.
Where do you live, Laura?
She's trying to say Sheffield.
And how old are you?
So Laura is a 4-year-old child who has this heterozygous mutation of FOXP2, which, by the way, regulates SLIT1 as well. And she has difficulty producing learned speech and learning how to produce it, but her auditory learning, auditory comprehension is pretty good compared to her speech production. You see a common theme going on here? And so working with Simon Fisher, we created mice with Laura's mutation, and we looked at their ultrasonic songs, and we noticed, right, even before the mutation, mice tend to sing more complex sequences of syllables when there's a female around, and more simple ones when they just smell female urine. And we find in wild type here, that's what you see, this increase in these more complex songs. The FOXP2 mutant mice had difficulty switching to these more complex sequences, shown with the white bars here. Kind of like Laura, they have difficulty sequencing complex sequences of phonemes together. And what about those layer 5 neurons that project to nucleus ambiguous in the mice? Well, they are there in the FOXP2 mutant mice with Laura's mutation. However, instead of being primarily localized in the motor cortex region, they're more spread out in frontal cortical areas here, shown in red. So we think FOXP2 is responsible for localizing these laryngeal motor cortex neurons to the proper pace in the cortex. And so even though these vocalizations are innate in mice, even though they can change with some low level of plasticity between different social contexts, we think that FOXP2 is already being used for vocal communication of innate sounds, and speech just depended upon it even more than innate vocalizations. And is this ethologically relevant? Yes. If you give a female the choice of these more complex, what I call sexy songs, and these more simple ones, and put her in a Y maze, she spends more time listening to the sexier songs, of course. And so we think that makes a difference. And so to bring it all back home here, what the take-home message is that even though songbirds and humans are separated by 300 million years from a common ancestor, convergence brings you about some interesting parallel systems where you can make hypotheses and predictions across these species, down to the individual cell types here, the individual connections going from one brain region like HPC and RA here, and here on the outside I have the specific cell types in the speech areas of the human brain that I argue is parallel to what these songbird circuits are like, setting up this hypothesis. And from a bigger evolutionary perspective, if two different lineages of vertebrate species can involve similar brain regions with similar connections to peripheral motor neurons for control of a complex behavior like spoken language and vocal learning, I predict that another 500,000 or 5 million years from now, if a turtle or a crocodile or a fish or another mammal, a chimpanzee, for heaven's sake, could evolve a vocal learning behavior, this is what it's going to look like. And that there is a limited way in which the brain is allowed to evolve these circuits. And one day, by figuring that out, we can figure out why can't our closest relatives say apple, and maybe even genetically modify them to be able to say golden delicious and so forth. And so the take-home messages here are that complex behaviors can evolve multiple times, even something like spoken language ability, from deeply homologous but diverse brain circuits, cells, and genes. And a trait like spoken language can be understood by studying very distantly related animals. And it looks like convergent mechanisms of a trait is associated with similarities and disorders of that trait. So even though there wasn't homology, but it's analogy, speech disorders in humans and equivalent disorders in birds seem to be similar. And so I will stop here and thank all the people that contributed to this. It really takes a big village to do all of this work. I named their names throughout. This is what my lab was back in 2017, right before the pandemic. I have to update the picture. And the funders that funded me in the last several years for this project and the VGP project from Howard Hughes, the Keck Foundation, and particularly NIH Director's Transformative Research Award. I couldn't found this mouse project with a typical R1 grant. Forget about it. It was too ambitious, it was crazy, and so forth. But a transformative award, they just put that name in front of it, and suddenly it could. All right, so I will stop there and take any questions should there be time. Thank you for your attention.
Microphone's on the way.
Great. So with that microphone, they could hear online as well, right? Yes.
I think so.
Okay.
Great talk. I really enjoyed it. So the androgen receptor, that right? You said you wanted someone to ask you about that. So
Yes, thank you.
I thought I would oblige. So normally we always think about androgen receptors being activated by testosterone, but I'm friends with Jessica Tolkien. I'm sure you know her over at Cold Spring Harbor, and they talk sometimes about like glucocorticoids activating androgen receptors. And so I was just wondering if you thought about, since it, right, you mentioned the androgen receptor was in some of the vocal learning areas in the human brain. And,
That's right.
right, and so it's like hard to imagine, so why do you need androgens in women since they can speak also? So is there some other ligand activating it, and then maybe working in a way that we don't normally think about in endocrinology?
Well, that would be a new idea for me. I'll keep in my pocket and reach out to you one day if it turns out to be true. Yeah, I think that I can go along with that idea, perhaps, that there could be a different ligand than what we normally see, as opposed to testosterone itself. On the other hand, looking into this gave me a better appreciation of thinking about the evolution of vocal learning and any kind of sex differences, and so women do synthesize androgen, testosterone, so that does exist. And some have argued that spoken language first evolved for sexual selection. And typically you find these sex hormones functioning in the brain for sexually selected behaviors. And in the songbirds, actually vocal learning involved in both sexes first, before it was lost in females of some sexes. So females lost the trait, and when you look at those species where both sexes still have the trait, both of them have high androgen receptor binding site, receptors in their vocal learning nuclei. So I think maybe vocal learning first evolved for us to sing and track and mate with each other, like I was telling some people earlier today, like our Jennifer Lopez and Ricky Martin types, right? And then after that, it became used for more abstract communication, but still with some kind of mechanism that it involves development of the circuit, so you got to keep the androgen receptor there to specialize these genes. That's the hypothesis I have now. I'd be curious to hear if anybody else thinks, another one would work better.
Anyone want to take that up?
Anybody online too, if you want to type, I know there's somebody checking out the questions online.
Well, as a little follow-up, while Dan checks the questions, is it known whether the androgen receptor plays a role in the sexual differentiation process in those areas for the species that have sexual dimorphism in song?
I don't think it's the androgen receptor so much, more like the estrogen itself, estrogen, which I guess gets converted. Does estrogen get converted to testosterone?
Vice versa, yeah,
The other way
testosterone
around.
to estrogen.
The other way around, right, yes. So some of you know about this. Mark Kanishi a long time ago, that if you overdose young zebra finches with high levels of estrogen for a few days early in life, they do not lose their vocal learning circuit. And we are recently found, or actually no, we're about to submit a paper to show that this is going through the sex chromosome, the Z chromosome of one of the sex chromosomes of birds, in terms of the genes that are being blocked. So somehow development of the circuit became dependent upon high levels of estrogen. But if you block estrogen in a male or block androgen in a male, the vocal learning circuit still develops. So it's not like necessary. I think it's making the vocal learning circuit activatable for sex-related communication, as opposed to developing the circuit, right?
Okay, cool. Okay, other questions for our speaker? Anyone on Zoom? Oh.
Yeah, Megan has a question. You say Megan or Megan?
I go by Megan. Yeah.
Megan, okay, all right.
So I'm seeing mostly that you're finding genes that have changes in expression, like gene regulation. But the genes themselves, have they changed at the protein level? And do you find kind of positive selection in those species? And also like genomically, like what is the genomic changes? Are there duplications or structural variation that could be contributing to what you're seeing in the expression profile?
Yeah, so yeah, we've looked hard for protein coding sequence changes that could be convergent between vocal learners. And we do have some evidence for a few changes, actually one of them in SLIT1, it turns out to be, which is making us think maybe regulatory changes are driving amino acid changes, you know, some type of dual selection there. But the number of amino acid changes is a handful, and we don't see it between birds and human. So I think regulatory changes is turning out to be more the rule than the protein coding sequence changes in these vocal learning circuits. And the few that exist, we have to wait another year or two to make sure we tidy up those studies to really prove that it's convergence amongst birds. In terms of the actual nucleotide changes in the genome, whether single nucleotide changes like I showed you for the androgen receptor binding site in NeuroD6, or structural variants, that's what we're going to start looking at going forward with these new high-quality reference genomes. I can say this much, overall, the vocal learning bird species look like they have higher chromosomal recombination rates. There's more GC-rich biased gene conversion happening. There's more chromosomal rearrangements in parrots compared to all other birds. And so I think that could be related to it. And I'd like to hear from you or others whether or not humans, do we have more structural variation compared to other non-human primates? Would that be an interesting correlation to look at?
Yeah, I mean, the great apes definitely have more than other primates.
Okay.
But humans themselves don't have more than the other apes, like chimp and
Okay,
gorilla.
all right.
Yeah.
Oh, that's interesting.
But it's a start, yeah.
Okay,
Yeah.
all right. Way back there? Okay.
I was just thinking about parrots, and one of the amazing things about them is that they can learn so much human speech and use it in complex ways, but in the wild, they all sound like they're just screeching at each other. And I'm sure they're hearing more than we're hearing, but
Right.
but we still know very little about how all this plays out in the wild in parrots.
Right.
I'm curious if there have been comparisons between parrots raised in captivity talking to people versus those in the wild just interacting with other parrots, or if we'd expect any sort of developmental differences or...
Yes, good question. I think we humans overrate our abilities, including for speech, and I think we underrate non-human animals, with parrots being one of them, despite what great things we see them do. So in captivity, actually parrots learn best how to imitate sounds by not having didactic tutoring like you would have in a classroom, but like how human children normally do it, whereas parrot is observing two people speak, or observing a human and another parrot speak, and they want to get in on the action. And if they start imitating and you go, yay, and start clapping like yay, you know, like a little child, right? And pet the parrot and so forth, you reinforce that learning, and that's how the parrot learns, through social reward, basically. And I think that is what's going on in the wild. There's social reward and also mating reward. It seems as if parrots who, including like African greys, they're like a mockingbird, they're copying many different sounds in the environment. And the more they can build up their repertoire, the more intelligent they seem, and the more other birds they attract to them. It has been argued by some, like some of these, there's a green parrot in India, where they just duet, and who are also in the wild and in captivity, they talk to each other in human speech, right? And this seems to be something that's possible and understudied. And so someone needs to go out to the wild and figure out what's going on. Do I think they have a full-blown language? No. But just imagine yourself plop yourself as a 10-year-old child or even 20-year-old adult into let's say you've grown up, I don't know, you've grown up in North Carolina and you never met another person who speaks another language for the first 20 years of your life. Go to China and hear people talking and think about that. Well, that's going to be your experience with animals. It's going to take you years to figure out what they're saying. I don't know if that analogy works well enough, but that's the one I can think of.
Any last questions?
Oh, yeah, so it's amazing to me how convergent, the level of convergence you're getting between these very evolutionarily distinct lineages. And you showed the example from the attack-seek data where you found a particular enhancer that's likely independently accrued changes on these lineages.
Mm-hmm.
Do you have a sense of whether you think the genetic changes are mostly convergent or not? Because surely they can't all be. You might have convergence maybe at the network level, but the individual genetic changes or molecular changes will be different. Are you starting to find any leads?
Yeah. What we're seeing a hint of so far is that the more closely related the species, the more likely to converge on an identical nucleotide or amino acid sequence. Mm-hmm. The more distant related, the more likely to converge on the same gene, but somewhere different in that gene, mm-hmm. that will have a similar functional consequence. So for example, songbirds and parrots have more convergence, higher level of shared convergence with each other than songbirds do with humans. It doesn't mean that there aren't any genes that parrots are more similar to humans than songbirds. That is the case, but this seems to be a general principle. So I think we're seeing both, and that is influenced by how close you are. And why would that be the case? I think it's because selection is a dual interaction between the structure of the genome, the structure of the cell, the functions of what's going on in the cell, and the surrounding environment. And so the two need to work in concert. So if you begin from similar humble beginnings, it's going to be easier to converge to the same solution than if you begin from very different beginnings. You see what I'm saying? Yeah.
All right. I think we've had Dr. Jarvis work extremely hard.
No, this was fantastic.
Today.
Thank you very much.
Yeah.